Hello, good morning students. Today we have to start with a new topic which is known as gynecium. As we all know that gynecium is the female reproductive part. It may have a single pistil which is known as monocarpillary. There is a single pistil, then it is known as monocarpillary, or more than one pistil, then it is known as multicarpillary. See, in multicarpillary, the pistils may be fused together. Okay, if the pistils are fused together, then it is known as syncarpus, and when it is free, then it is known as apocarpus. You can see in the diagram that A is a multicarpillary. Okay, all the pistils are fused together, are free. Okay, now in the B diagram, it is known as multicarpillary. The syncarpus pistils of papaver means all the pistils are joined together. In the third figure also it is mechelia, in which you can see that many carpels are present. Okay, but all are free. You can see in the diagram. Okay, now next we are going to see the structure of a flower. In gynecium, each pistil has three parts. In gynecium, each pistil has three parts. One is stigma, style, and ovary. Stigma, style, and ovary. Now, the main function of stigma is to capture the pollen grains. Okay, that's why it is written landing platform for pollen grains. Second one is style. Style, elaborated cylinder part beneath the stigma. It is a long tube-like structure which connects stigma with the ovary, and ovary is the basal part, okay, in which the ovules are present. Now here you can see this is the structure of stigma, style, and ovary. Below is the ovary. Okay, this is the complete structure of a flower, reproductive part of the flower, gynecium. Okay, now in gynecium, inside the ovary is a ovarian cavity. Ovarian cavity is also known as locules, in which placenta is located. Means many ovules are connected together with the help of a structure which is known as placenta. Okay, now with the help of this placenta, ovules are connected. These ovules are known as megasporangia. Okay, I hope you remember the ovules. Another name is megasporangia, and pollen grains, another name is microsporangia. Okay, now the number of ovules in an ovary may be one, for example in wheat, paddy, mango, etc., to many like in papaya, watermelon, orchids. Okay, so the number of structures and number of ovules in an ovary vary from species to species. Okay, you can see in this diagram, this is the structure of a typical ovary. Okay, you can see that placenta is there, and attached to the placenta is a funicle. So funicle is a connection link between the placenta and the ovule. Okay, now we are going to see in detail the structure of megasporangium. Structure of megasporangium or the ovule. Structure of megasporangium or the ovule. Now this ovule is attached to the placenta by a stalk which is known as funicle. Ovule is attached to the placenta by a stalk which is known as funicle. You can see in this diagram, funicle. This is the funicle. Now, the junction between the body of ovule and funicle is called hilum. Okay, so hilum is again a tube-like structure which connects the funicle with the ovary. Okay, ovule of the ovary. Each ovule has one or two protective envelopes. These protective envelopes are known as integuments, means the covering layer is known as integument. These integuments are absent at one place, that is known as micropyle. Okay, that end is known as micropyle, and that from that point only the pollen tube enters inside the ovule. Okay. Now, opposite to the micropylar end is the chalaza or the basal part. You can see in the diagram here. The lower part is known as chalazal part. Chalazal part is the opposite to the micropylar end. Now, see in the diagram, the outer wall is the ovary wall, then placenta is there. After that, a connection link which is known as funicle, ovule, and locule. Locule is the cavity. Okay, locule is the. Now you can see the structure of megasporangium in detail. Okay. Now enclosed within the integument there is a mass of cells which is called as nucellus. Okay, it is called as nucellus, and nucellus is a reserve food material. Okay, in this diagram of ovule, you can see the structure of a typical seed. A typical seed. Now, inside the nucellus is the embryo sac or the female gametophyte, which is known as embryo. An ovule generally has a single embryo sac formed from a megaspore. From a megaspore, single embryo sac is present. Now inside this embryo sac we can see many female gametophyte. Inside this embryo sac we will be able to see the female gametophyte. Okay. Now the process of megasporogenesis. Megasporogenesis is after fertilization, the development of the embryo sac. Okay. Now, it is the formation of megaspores from megaspore mother cell, the mature megaspore mother cell. This process is known as megasporogenesis. Maturation of the ovule is known as megasporogenesis. Ovules generally differentiate a single megaspore mother cell in the micropylar region of the nucellus. Now, one ovule is having a single megaspore mother cell, and that megaspore mother cell is found in the micropylar region of the nucellus. It is a large cell containing dense cytoplasma and a prominent nucleus. In this, megaspore mother cell is a very large cell which contains dense cytoplasma and a prominent nucellus. Now, nucellus provide nutrition to the embryo sac. Okay, and mature mother cell undergoes meiosis to produce 4 megaspores. It produces four megaspores. That's why it is known as tetrad. Okay, at the micropylar end, nucellus is there and megaspore mother cell. Now, this megaspore mother cell will divide first into dyad. Dyad means 2, and then into tetrad means 4. Okay, this division takes place inside the embryo. Now, formation of female gametophyte or embryo sac. How this embryo sac is being produced? Now, in majority of flowering plants, one megaspore is functional while the other three degenerate. Just now we had seen that tetrad is being formed, megaspore tetrad, and from this megaspore tetrad, only one is the functional one, while three other will degenerate. Okay. The functional megaspore develops into the female gametophyte, and this functional megaspore means the one which is growing, which is developing. Three are degenerated and one is the functional. The embryo sac formation from a single megaspore is called monosporic development. Okay. Now this embryo sac is formed from the single functional megaspores. Since it is single, then that's why it is known as megasporo. It is known as monosporic development. Okay. The embryo sac formation from a single megaspore is called monosporic development. Now you can see the step-by-step development of the embryo. Okay, mature embryo. This is, we are going to see formation of female gametophyte or the embryo sac. Now the nucleus of functional megaspore divides mitotically. Okay, mitotic division will takes place to form two nuclei. They move to opposite poles, forming two nucleate embryo. You can see in the first diagram, it is a two nucleate embryo sac. After that, it will become four nucleate, and these four will divide mitotically and form eight nucleate stage. Eight nuclei are there. Okay. Now the nuclei again divides two times, forming four nucleate and eight nucleate stage of the embryo sac. This division are free nuclear, that is, nuclear division are not followed immediately by cell wall formation. Means a new embryo is not being formed by forming a cell wall. Okay, this is the typical structure of a embryo sac. At micropylar end, we are having two synergid cells and one egg cell. In the center, we are having two polar nuclei, and at the base, we are having antipodal cells. We all remember that these two synergid cells will provide nutrition to the egg cell. Okay. Now, after the eighth nucleate stage, the cell walls are laid down, leading to the organization of a typical female gametophyte. After 8 nucleate stage, the cell wall is formed. Okay, and it will form a typical female gametophyte. 6 to 8 nuclei are surrounded by cell walls and organized into cells. Remaining two nuclei or polar nuclei are situated below the egg apparatus in the large central cell. Two polar nuclei is seen in the large central cell. A typical egg cell is having a structure. See, at the chalagial end, we'll be able to see the antipodal cells, and at micropylar end, we are having one egg cell and two synergid cells. There is one filiform apparatus. Okay, this filiform apparatus is to hold the synergid and the egg cell, and it also guides the pollen tube towards the egg cell. Okay, its main function is to guide the pollen tube towards the egg cell. Okay, now distribution of cells in the embryo sac. How many cells are there? A typical mature embryo sac is 8 nucleate and 7 celled. 8 nucleate and 7 cells. You can see here. At antipodal end, there is 1, 2 and 3. In the middle, this is polar nuclei 1 and 2, 5 total, and in the synergid cells we are having 3. So 5 plus 3, 8. Okay, so it is 8 nucleate and 7 celled. Now why it is known as 7 celled? Because the polar nuclei will combine together and it is considered as one cell. Okay, now 3 cells are grouped at the micropylar end and form egg apparatus. It consists of 2 synergids and 1 egg cell. You can see here. Now, synergids have special cellular thickenings at the micropylar tip. It is known as filiform apparatus, and this helps to guide the pollen tube into the synergids. And 3 cells are seen in the chelagel end. They are known as antipodal, and the center contains 2 polar nuclei. So total there are 8 nucleate and 7 celled stage embryo sac. Okay, now formation of female gametophyte or the embryo sac. See, mitotic division takes place. After that, the functional megaspore mother cell will undergo 4 stages, for different stages. Means here the meiotic division will take place. Now at the end we are having this mature female gametophyte or the ovule. I hope students you had understood what we had seen just now. See, learn all these concepts, these names from the book, and we are going to meet again in our live class in the next live class. Till then, goodbye, take care.